based on phylogenomic analyses. So here we use whole transcriptome data sets to reconstruct the evolutionary relationships among 14 representative species in the group. And together with a previously published fossil calibrated molecular phylogeny of the family, we estimated that Jaltomata diverged from Solanum within the last 17 to 20 million years. Jaltomata itself diversified within the last 3 million years, and this particular clade down here, which encompasses the most species in the genus, as well as the most just broad floral diversity, diversified within the last 1 million years or so. All right, so what are potentially some genetic and or developmental processes that could have facilitated this apparent rapid change? And one way that we have looked at this is doing a comparative genomic approach. So in other words, is there something in particular about the genomic structure in Jaltomata that may have facilitated such rapid change? And one of the interesting things that we found is that there is significant expansion of relevant gene families, specifically on the branch leading to Jaltomata. So right here. Obviously, we also detected expansion and contraction of gene families in other Solanacea species that we looked at, but we were especially excited because many of these gene families are known to function in floral development and regulation of meristems. And so these provide really exciting targets for follow-up work. And so we do have some hints that potentially differential expansion of gene families may be playing a role, but this comparison is still at pretty large evolutionary timescales. So narrowing in to Jaltomata, I examined the floral development of 5 representative species, as well as a wild tomato group, to try to figure out developmentally what's going on to give our divergent mature floral traits. So where and how does floral development differ to give rise to these different mature forms? Here I'm showing you the developmental trajectory for just one of our species we looked at. And it was our nice little tubular species. But in general, what we found is that it is differential growth rates of particular floral organs and specific regions of those floral organs that give rise to these different mature shapes. And in particular, we found that these changes emerge in later stages of floral development. And it has been predicted that such changes in later stages are easier to evolve rather than changes in the beginning of floral development, which might have more negative fitness consequences on flowers. All right, so I've given you some hints as to potentially genomic and developmental reasons why we might see rapid change. But what about the genetics? In particular, among really closely related species that have highly divergent floral traits. And for this, I want to characterize the underlying genetic architecture between these divergent floral traits. In particular, how simple or complex is this genetic architecture? So we might predict that traits with more simpler genetic bases might be easier to change. Does the complexity of these traits differ between the type of trait that it is? And finally, how pervasive and in what direction is genetic plytropy, which is when one gene influences more than one trait. And so we might expect that if it's antagonistic, such that a single genetic change influences more than one trait, and those traits then have opposite fitness effects, this might slow the rate of change in those traits. In contrast, if the plytropy is adaptive, such that a single genetic change affects more than one trait, and this has all-around positive fitness effects, we might expect to facilitate change in those traits. To address these questions, I did a classic QTL mapping approach where I generated a segregating hybrid population between 2 species that are close related but show divergent floral traits. And these two parents are actually ones that I included in my comparative developmental work as well. And this Spinosa is the species that we now have a genome for as well. All right, so I phenotyped a whole bunch of floral traits, including your standard morphological ones, as well as inflorescence size, the volume of nectar produced per flower, and I also quantified petal and nectar color as well. Further, I also phenotyped several fertility traits. And this is because in a previous study, I found substantial yet incomplete reproductive barriers between the two species, and so I wanted to better understand the genetic basis of those traits. Also, there's some evidence to suggest that genes involved in floral development may also function in seed and fruit development as well, or interact with genes that do. And so this potentially could have been another source of pleiotropy influencing our floral traits. Okay, so I measured a whole bunch of traits. Here are some of them here. And I assure you that most of them did in fact significantly differ between our two parents. It's a good thing, we need that for QTL mapping. Okay. When we look at the segregation of our traits in our population, this can inform us of the mode of inheritance for the genes underlying our trait. So in this particular example here I'm showing you is style length. So on the y-axis we have the number of individuals that show style length of a certain value. So here in the blue we have one parent, and the purple we have the other parent. Green is the F1s, and the red show the distribution in our backcross individuals. So this pattern right here, where we have divergence in the two parents, the F1s intermediate, and the backcross ones unimodally distributed with a mean in between the F1s and the recursive parent, this is indicative of primarily additive effects underlying our trait of interest. And in fact, we found this pattern consistent with primarily additive effects for most of our floral traits that we examined. In addition to looking at patterns of segregation, we can also look at correlations among our traits to inform potential genetic associations. So I did in fact recover significant associations within trait categories, so among certain aspects of morphology, among certain fertility measures, but generally not between different types of traits. The exceptions being associations between some measures of floral morphology and both nectar volume and color. And this survived correction for multiple testing. All right, so I showed you that segregation and correlations indicate that it potentially could be relatively simple with few antagonistic relationships. What do the QTL say? So here we used genotyping by sequencing to get markers and genotype our individuals, and also to construct the first linkage map in the system. We were able to identify QTL for most of our traits. In fact, I found between 1 and 4 QTL for most of our traits. Further, most of our traits also had at least one QTL with moderate to large effect. And in many cases, when you summed the effects across all the QTL for each trait, it explained the majority of the parental difference, indicating that we recovered at least most of the QTL underlying that trait. Obviously, without QTL mapping, we're not going to recover all of them, but this suggests that we at least recovered most of them. Looking at the distribution of QTL across the genome, here I'm just showing you a subset of the traits. If I showed you all of them, this map would be quite overwhelming. We did in fact identify QTL on all of our chromosomes, but as you can see, we did identify some nice little clusters on certain chromosomes. And as suggested by our correlation analyses, these correspond to overlap among traits within certain categories. So among different morphology traits, among different fertility traits. We generally did not find QTL overlap between different types of traits. The exceptions being among certain aspects of floral morphology and both nectar volume and nectar color, like we saw in the correlations. So we were actually really excited about this because this association between larger flowers with darker and more nectar is seen consistently across the genus, seemingly convergently so. And we think it also might be associated with transition to hummingbird pollination. And so if there is an association on the genetic level, this may have facilitated this change that we see across the genus. All right, so going really fast now. I generally found a relatively simple genetic architecture for many of our traits, a general lack of antagonistic pleiotropy, and potentially a case of adaptive pleiotropy. And so overall, we see potentially differential growth of gene families, changes in later stages of floral development, relatively simple genetic architecture, and general lack of antagonistic pleiotropy with some potential adaptive pleiotropy that may all have contributed to the extensive variation that we see in the genus today. With that, I'd like to thank my advisors and collaborators in funding, and I'll take questions.
So what's the rationale of using the bycross for future marginal as a tree?
Because that's the only one I got to work. Excuse me, the first one I got to work. Yeah. Yes.
So you said hummingbirds might be an important selective agent. So
I haven't tested it.
so, but could it be that the correlations you observe are those that are filtered after selection, and rather than being a mutational bias that facilitates this evolution, what you're seeing is actually the outcome of selection.
Is the outcome of selection.
Outcome of selection, yeah.
So we know that the ancestor of the genus was rotate and presumably bee pollinated. I can.
Okay.